Hello and welcome to another Mr. Carter Science Special. In this video we'll be looking at a brief history of bacteriology. One of the common themes that runs throughout the B5 unit of the AQA Biology course is the idea of preventing infection and preventing the spread of infections. Well, most of our current methodology relies heavily on the research of a number of scientists from both the 1800s and the 1900s, the 19th and 20th centuries. So we're going to have a look in this video at a number of those scientists and how they've contributed to our current methods that will prevent the spread of infectious disease. The first person we're going to mention is Ignaz Semmelweis, and he was a Hungarian scientist working and studying a disease known as childbed fever. And Semmelweis studied cases of childbed fever in his hospital where women died after childbirth. In his hospital, people giving birth were helped either by midwives or by student doctors or doctors themselves. And Semmelweis compared the rates of childbed fever in each of the groups. And there was a lot higher rate of childbed fever in the wards that used student doctors. The doctors and student doctors would often go and dissect dead bodies and then go directly to help deliver a baby. Semmelweis thought that they might be carrying the cause of childbed fever with them. And he insisted that the staff at the hospital wash their hands using a weak chlorine solution. The cases of childbed fever in his hospitals were reduced by hand washing and good hygiene. Unfortunately, his conclusions and his ideas were ignored for a very long time. He is one principal person on your course you should have come across already, but there are many others that you may not be aware of. We can go back in this history of bacteriology all the way back to 1670s, and we're going to go through each of these 7 scientists and see how they've contributed to our understanding of disease and the prevention of and spread of disease. So the first scientist, Antony van Leeuwenhoek, he is the first person in the 1670s to see bacteria using a microscope. Remember, bacteria cells are much smaller than animal or plant cells. Almost over 100 years later, Edward Jenner in 1796 proves that the disease cowpox prevents smallpox infections. Remember, smallpox is a highly dangerous viral disease that kills and disfigures huge numbers of people at that time. But if you've been infected with cowpox first, then you will not be infected with smallpox. And this is the first vaccine. In fact, cowpox was known as vaccinia, so Edward Jenner used the word for his new invention, the vaccine, and we still use vaccines today. In 1847, as we've already discussed, Ignac Semovice in Vienna proves that washing hands reduces the spread of infection. In this case, he was studying the childbed fever disease. But his ideas are rejected due to a lack of scientific explanation. People at the time, scientists at the time and doctors at the time thought that childbed fever was God's punishment against women. Then later on, in 1862, Louis Pasteur disproves the spontaneous generation of bacteria, and he proves and develops the idea of germ theory. And he does this by using swan neck flasks. At the same time, he invents something called pasteurization to heat and destroy microbes, and this prevents the spoilage of both beers, wines and milks. And indeed, pasteurization is still used today to prevent milk from going off so that we can keep it for much longer than you would be able to with just fresh milk straight from the cow. So here is Louis Pasteur's experiment which disproves the spontaneous generation of bacteria and microbes. He took a swan neck flask, and you can see here this is the neck of the flask. It's a glass apparatus which has a particular curve on the neck, and this particular curve means that air can enter the neck, but it is unlikely to go up and down and into the actual flask. So he began by sterilizing his flask and the broth that it contained, almost a soup that it contained, by applying heat, and he lets the flask sit, and there's no bacteria present many days later. In his second experiment, he does exactly the same thing. He's got the same broth in the same flask. He applies heat and sterilizes the broth, but then he removes the swan neck and he lets it sit. 2, 3, 4 days later, he discovers that we have bacteria present. In the third experiment that he does, he has the same apparatus again, same broth, he applies heat again, but this time he tilts the flask sideways and lets it sit. So he gives a route for air and any airborne bacteria to flow through the neck, and again bacteria are present. At the time, people thought that bacteria would just spontaneously appear out of nowhere. If that was the case, we would expect to have bacteria in this flask in his first experiment, but that's not true. It doesn't happen. So Louis Pasteur disproves the idea that bacteria can spontaneously generate. And this leads us on to think about how bacteria can be spread from one person to another person, or spread through the environment. Around the same time, in the 1867s, Joseph Lister in Glasgow builds on the work of Pasteur and uses carbonic acid to wash hands and instruments and rooms, and reduces the case of infections, in his case a disease called gangrene, after surgery. So he invents one of the first antiseptics and he uses it in his operating theatres. He had similar ideas to Ignaz Semmelweis. In the 1870s, Louis Pasteur again works on diseases in chickens and artificially produces a vaccine against chicken cholera. He later develops an artificial vaccine against anthrax, again building on the work of previous scientists. In the 1880s, 10 years after Louis Pasteur has done this work, Robert Koch isolates the anthrax bacteria and proves it actually causes the disease. Up until this point, there was confusion as to whether the anthrax bacteria caused the disease or perhaps cholera caused the disease anthrax. So Koch builds on this and identifies what's known as 4 postulates. And he proves that cholera and TB are caused by different bacteria and wins the Nobel Prize for this. And then in the 1820s, 1828 to be specific, Alexander Fleming, while working on a staphylococci bacteria, gets a fungal contaminant in his petri dish, and he discovers the world's first antibiotic, the world's first chemical that we can harness to destroy bacterial cells. So let's just go back a second and look at Cox postulates. For a scientist to confirm that a microbe causes a disease, the following conditions must be met. 1. The organism must always be present in every case of a disease. 2. The organism must be isolated from a host containing the disease and grown in a pure culture within the laboratory. 3. Samples of the organism taken from a pure culture must cause the same disease when inoculated into a healthy, susceptible animal in the laboratory. 4. The organism must be isolated from the inoculated animal, and it must be identified as the same original organism first isolated from the original diseased host. If those 4 postulates, 4 conditions are met, then you can confirm that a microbe causes a disease. Here's Alexander Fleming, and he has an extraordinary quote. I'll read it to you. When I woke up just after dawn on September 28th, 1928, I certainly didn't plan to revolutionize all medicine by discovering the world's first antibiotic or bacteria killer, but I suppose that's exactly what I did. Before this moment, bacterial and viral diseases, but in particular bacterial diseases, were killers and were rightly feared across the world. Now, diseases that we might get ourselves, we no longer worry about because we can receive antibiotics from our doctor, which will kill the bacteria within our body and help us to overcome the infection, directly due to the work of Alexander Fleming and the other scientists we've mentioned already. The work of these scientists has led directly to improving our ideas about hygiene and the effectiveness of hygiene at preventing the spread of pathogens and preventing infections. Hygiene encompasses 3 or 4 different areas. For example, most importantly, hand washing, especially after using the toilet or before cooking and after contact with animals or contact with somebody who's ill, will destroy and prevent and reduce the spread of pathogens. Using disinfectants like bleach or Dettol on kitchen surfaces or toilets reduces the number of pathogens. Separating raw meat from cooked meat or separating raw meat from food that's eaten uncooked will prevent the spread of pathogens. For example, not letting lettuce, which you will eat uncooked, touch raw chicken, which is contaminated with a large amount of pathogens and needs to be cooked and heated up to destroy those pathogens before you consume it. In the agricultural businesses, maintaining the hygiene of people and agricultural machinery will help to control the spread of plant diseases, which is very important if we're looking at farms or hydroponic farms, or indeed cultivation of flowers for sale. Other ways that we can prevent the spread of communicable diseases that build upon the work of the scientists we've already mentioned in this video include isolating individuals, quarantine. This is something we've all become used to during the current pandemic of COVID-19. If somebody has an infectious disease such as Ebola or cholera, then keeping them in isolation will prevent the spread of the disease to healthy people. Destroying or controlling vectors. Many diseases are carried by vectors like mosquitoes, rats, flies, fleas, aphids. Destroying the vectors reduces the spread of these diseases. They cannot be transmitted from one person to another if the vectors do not exist in that environment. Places like Florida or Southeast Asia often will try and destroy mosquito vectors for malaria by using pesticide chemicals and spraying a fog of these pesticides over areas where mosquitoes are known to breed. Vaccinations. A vaccine is an injection of dead or altered pathogens. This builds on the work of Edward Jenner, and these dead or altered pathogens will cause your immune system to produce antibodies, which will protect you from the real pathogen. Vaccinations can protect a large amount of people from diseases like measles and today COVID-19. Vaccines cannot protect plants from diseases, however, because plants do not have an immune system. But if we get a large enough number of people in a population vaccinated, we might be able to achieve herd immunity. And that means that the pathogens will not be able to spread from person to person, and therefore people who cannot receive the vaccines for medical reasons, or people with compromised immune systems due to organ transplants or old age, or indeed newborn babies, will be protected from the pathogens because our population has reached herd immunity. I hope you've enjoyed this brief history of bacteriology. I feel that it's very important that we understand the names and the contributions and the successes of scientists who've gone by 100, 200 years ago and have contributed to the ability that our current generations have to not be afraid of diseases anymore. I feel it's a very important thing for you to understand that. I'd love to see your comments on this topic, so don't forget to write in the comments section below. Please like and subscribe so you'll get notifications of when I release the next videos. Thank you very much for your time.